This is a study on the effects of caffeine on cortisol excretion by Cole Fisher and Chaz McElroy.
Some background on cortisol is it's basically a fight or flight similar to adrenaline, and previous studies that we found is that it can be induced using caffeine.
The biological process of excreting cortisol starts when the hypothalamus senses that the body's blood sugar is low and it releases the hormone CRH. CRH is then sent to the pituitary gland, which triggers the release of ACTH, which is then sent to the adrenal glands, and cortisol is then released.
The purpose of our experiment was to find the amount of time it takes cortisol to show up in different body fluids after caffeine is consumed.
Our hypothesis was that cortisol would show up first in blood, then saliva, then urine.
This is a table showing how we added the samples into our plate. For each sample, we used triplicates.
Our subject was an 18-year-old student. We chose someone who didn't consume caffeine regularly because we thought that would give us a bigger fluctuation in the results. Therefore, it would be easier to see what happened. Blood was drawn by a trained phlebotomist at a local clinic, and it was put in test tubes specifically used to separate the plasma from the red and white blood cells. The subject was asked to urinate in a 50 milliliter sterile conical and was asked to spit into a 1.1 milliliter sterile epitube. After each sample was collected, they were put on dry ice so that we could safely transport them while maintaining the integrity of the samples back to our lab.
The ELISA kit we used to analyze our samples had 7 calibrators with the concentration as follows: 0, 0.5, 1, 5, 10, 20, and 100 nanograms per milliliter. This is the procedure from the assay kit that we used and what we added into each calibrator, sample, and blank well. We added 25 microliters of calibrator into the calibrator wells, 25 microliters of sample into the sample wells, as well as 200 microliters of diluted conjugate into both the calibrator and sample wells. We incubated for 37 degrees for 1 hour and removed the contents and washed 3 times with 300 microliters of diluted wash solution. After washing, we added 100 microliters of TMB substrate to all 3 calibrator, sample, and blank wells and incubated at room temperature in the dark. We then added 100 microliters of stop solution into the calibrator, sample, and blank wells, shook gently on the microplate, and read at 450 nanometers.
This is a table showing the average absorbance numbers of cortisol levels in the blood at each time period.
This is a table of the averages of all the samples in saliva at each different interval.
And this final table is similar to the last ones. It shows the absorbance levels for each time that the samples were taken at for urine. This is our sample t-test table. The top number is the mean, which is the average from each of those triplicate samples. The variance is the standard deviation within that sample from each one of the triplicates. The t-stat is a number similar to standard variation, which just shows how reliable a sample is within itself. And then the two-tail p-value shows how likely it is that that sample came simply from chance.
These are the t-stat values for the remainder of our samples. So, restating our hypothesis that cortisol will show up first in blood, followed by saliva, followed by urine, and our data did not show any significance in the amount of time it takes, and the t-test showed that the null hypothesis was true.
Our findings indicate that there was no significance between the amount of time it takes cortisol to show up in blood, urine, or saliva. A possible reason for the null hypothesis was that we used a cortisol saliva kit and not a urine or serum kit as well. We didn't do this because it was much more expensive at $200 a kit, and our instructor said it would probably be safe just to use that. Another possibility for the null hypothesis is user error.
Further research we could have done was to use the urine and serum kits instead of just using the saliva kit. Another improvement would be to decrease the amount of time between samples.
Our teacher Henry Weeks helped us come up with the idea for this project. One of the people who helped us the most outside of our teacher was Jessica Elder, our school's athletic trainer. She helped us figure out the legality of working and drawing the blood. Dr. Chris Yarder allowed us the lab space and the help from the Cascade Family Clinic's lab technician, Tammy Carey, to help us actually get the blood drawn and a good space to be able to take the samples and get them safely back to our lab.